Good morning. Nice of you. So it is 10:40. I did say that I would give you a little bit of time to fill out the eval. By show of hands, is anybody actually going to do that right now? Maybe? Yeah? So how about I give you like 5 minutes? So I don't have a physical eval. You should have an electronic device with which to fill that out. So if you want to go ahead and you haven't already done so, let's take, 5 minutes or so to start digging in. I would really appreciate it if that's possible. Okay, so I'll give you 5 minutes. So you guys, if you've already done it, you can talk amongst yourselves, which you're already doing. Okay, so that was 5 minutes. So I'm going to start talking now. If you're filling out an eval, you can continue to do that. I'm just going to remind everyone of some of the logistical things that you should be reminded of. Today will be my last lecture to you guys of the quarter, so I'm going to make the most of it. Pretty pumped. So I'm looking forward to today's lecture. It's been a lot of fun this quarter. On Friday, Taryn and Christina, so you should have received an announcement that came from them regarding they were trying to poll you guys to see if there are specific topics. So kudos to them for soliciting feedback. Taryn saying specifically Christina did this. So if you have specific areas that you want covered on Friday, I will throw in the caveat that depending on how exhaustive that list is, they may not be able to get through everything in 1 hour session, but they'll do their best, okay? So it's just another opportunity to review some material. So that's on Friday. Our exam is on the 6th, which I believe is Tuesday. Is that right? What is that? Am I right? Tuesday. Give me some positive affirmation there. I want to make sure I'm not giving misinformation. So Tuesday the 6th from 4 to 7 p.m., so a different time of day than we're used to meeting and thinking of biochemistry. So make sure to note that on your calendars if you have not already done so. I have, again, reserved Oaks 103 as a satellite room right next door to free up some space in here. So again, we will have a situation where we have about 20 seats in that room to loosen up the seating in this room a little bit. Please, please, please remember to bring a pink Scantron, a pencil, and each exam there have been a couple of people who have forgotten a calculator. So please remember to bring a calculator with you, okay? You can expect the format of the exam will not be unfamiliar to you, okay? It will look very similar to the exams you've already taken in this class. It will just be longer, okay? We have 3 hours technically to run the exam, which is longer than I expect for sure the exam to take, but that's the allotted amount of time. The exam is essentially double the time of the exams that you've taken to this point, which really puts us more at a little over 2 hours, okay? At least targeted, all right? But for those of you who want and need extra time to the 3 hours, you will have that time. And what else can I say? I'll just remind you also that the final is cumulative, of course. It will review material that we've covered on exams 1 and 2, but the majority, or 50%, of the exam will review the material that we have not yet covered. And from a perspective of point value toward your final grade, for those of you who may not have done necessarily as well as you had hoped on one of the first exams, the final really provides an opportunity to come back, in essence, right? Because this exam is worth more than any of the other exams, okay? So that's not meant to put additional pressure on you for this exam, but rather because some of the material is reviewed, hopefully we can learn from having been tested already once on a bunch of that material, figure out why you got certain things wrong, straighten that out, and then revisit that material, okay? So are there any logistical questions at this point? No? Okay, you guys look ready to learn, so let's do that. So today, what we're going to do is finish off our discussion of really the material covered in chapter 28.2 of our book, right, which is a more detailed discussion of gene regulatory strategies, okay? And we're going to pick up with our discussion of the lac operon, which is sort of the, both for historical and conceptual reasons, the paradigm that we like to start with in biochemistry classes at the undergraduate level in discussing how gene expression can be coordinately regulated. And so we've already talked a bit about that, but we'll finish that. And then we'll transition to some other examples, particularly in bacterium, where there's, again, strategies for coordinated regulation of different gene classes. And what you'll see is that when we discuss some of these other examples, which will just be sort of a survey of some of these other strategies, it'll pick up on those general concepts that I opened the last lecture with, which is this idea of both positive and negative regulation, and how those general concepts can actually be put to work in a real example as opposed to just an abstract idea. Okay? Right. Okay. So where we left off at this point was discussing the Lac operon. And so hopefully now you're familiar with this idea of an operon being a collection of genes in the case of the Lac operon, the Lac Z, Y, and A genes that are all coordinately controlled or expressed under the control of a single promoter shown here. This is the more detailed diagram. Maybe I can zip back to here, let's say, to just make sure everyone's on the same page for what an operon is. So you have a promoter, you have these important operator sites which are going to bind to some regulatory factor. In the case of the Lac operon, that's the Lac repressor protein, as we discussed. And then you have also somewhere in the proximity of the promoter often, although I will mention just in passing that one distinction with eukaryotic gene regulation, which we're not going to really discuss in great detail, is that some of these activator sites can reside at very large distances away from the promoter, which is something that's sort of interesting about how eukaryotic genes are regulated. Yes? So the -10 and -35, which we discussed as a canonical set of sequence elements within the promoter itself, would be within this pink box of the promoter. Okay, those are elements that define the promoter itself. Okay, and then this operator or some other distal, adjacent site, those are sort of additional regulatory elements in the larger sort of operon structure. Okay, does that clarify that? Okay. So coming back to, we talked a little bit about DNA binding proteins. So coming back now to the main sort of challenge that is met by the regulatory strategies of the Lac operon is in addressing this question that we articulated, which is how does a cell both sense when there's a condition that requires expression of these genes, and I should also just remind you that these genes, of course, relate to enzymes and other protein factors required for using lactose as an energy source. So the question is, when should we turn these genes on? Well, obviously, in the presence of lactose makes a lot of sense, right? So you don't want to turn them on if lactose is not present because you don't need them, right? But then there's this added complication, which is that glucose, as I mentioned, is the preferred energy source of bacteria, which will shunt into the glycolysis pathway and be utilized as an energy source that way, as you'll learn in 100C. And so we really need to meet two criteria or two conditions to get this gene circuit turned on, which is that you should be in high lactose, right, for a sustained period of time. I'll come back to that idea in a moment. And you should also be in low glucose, right, so that you're not using the less favored energy source of lactose, okay? And so then the question becomes, well, how is that achieved, okay? So there are two essential components of regulating the Lac operon. One is this idea of the operator site bound to repressor, okay? So just to remind you, this is the repressor protein, which binds to the operator and physically prevents RNA polymerase from either binding to the promoter or acting at the promoter to initiate transcription. So that's like a roadblock, I think is the term I used last time. So that's an example of negative regulation in the context of those introductory concepts that we discussed. That's constitutively present. So it's basically always off unless otherwise told to be in another state. And that includes the case of when you are in a situation of low glucose and no lactose, okay? So when it's not sufficient to say, well, I'm lacking glucose to turn on this pathway, which makes sense, right, at first glance, because, again, you don't have lactose as a substrate to be broken down. But it does introduce the idea of positive regulation, which can occur also at the Lac operon. So when you're, which I'm going to describe in more detail in the next slide, but I'll just, for now, just review this idea that you have this additional protein, which is called the catabolite response protein or cyclic AMP regulatory protein. It goes by different names, but the basic function of it is to serve as a positive regulator of transcription at the lac operon. And so it's going to bind to some activating sequence, which is this purple thing that's hard to see in this looped diagram. And it's going to bind to this sequence in a cyclic AMP dependent manner. Okay? Cyclic AMP, I talked to you very briefly, becomes high, okay, when glucose stores are low. And again, you're going to learn the molecular details or biochemical reasoning for why that is when you learn more about glycolysis. But for now, we can just understand that relationship, that when glucose is low, cyclic AMP is high. And so what that means is that you have this small molecule. It's a modified nucleotide, as you might notice, cyclic adenosine monophosphate. And we call these small metabolic molecules that can relay signals about sort of the overall metabolic state of a cell. We call those often second messenger molecules. So cyclic AMP is a second messenger molecule, and it can bind to the CRP protein, and upon binding, activate it for positive regulatory function at the lac operon. Okay? But that is not sufficient, right, in this case, because we're under low glucose, so you have this positive regulator here, but you're still blocking. Your roadblock has not been removed. There's a question.
So the lac operon has both positive.
It sure does, yeah, which is one of the reasons why we teach it, right, because it invokes both of these concepts. Okay? Okay, so now let's see how those modes of regulation manifest themselves under a couple of other conditions. Okay? So now we have a situation. We're reverting back to the idea where glucose is high, but now lactose is also present. Okay? So again, coming back to this idea, if glucose is high, okay, cyclic AMP, it's inversely correlated with the glucose level, so it drops, right? So what that does is that means that this activator sequence, which should be purple, right, is no longer bound by the CRP protein. However, because lactose is now present, you're beginning to get the removal of the roadblock, okay, or the removal of this negative regulatory function of the lac repressor. I like to refer to that as de-repression, okay? And the way that that works is that the molecule called allolactose, which I haven't really discussed with you yet, so we call this an inducer molecule, okay? It's inducing expression of the lac operon by binding to the lac repressor and causing it to dissociate from that operator site, okay, and essentially removing the roadblock. So that's why we call it an inducer. It's interesting that allolactose is the inducer. So if I were to ask you just based on your intuition and first principles thinking, what might be the inducer of the lac operon? You know, I've already told you it's this molecule allolactose. What might you have said, right? Lactose, right? That's the very logical, that's what I would have said, certainly. But that's not actually the inducer. So that raises the question that maybe some of you in your reading might have pondered, right? Which is, why is it not lactose? And there's a reason for that, which I can't go into too much detail because it relates to enzyme kinetics. You can take that if you need to. No, I'm just kidding. It's the last class, you know. I can kid around, right? What was I talking about? Okay, so you're going to learn in 100B from my colleague Carrie Parch all about enzyme kinetics, right? And you're going to learn how enzymes, many of which we've talked about, right, in this class, respond to different levels of their substrate, okay, and how the rate of a reaction is impacted by the substrate level and so on. You're going to learn a lot about that stuff. Well, so it turns out that even when the system, right, let's say here, is repressed, right, there is still what we call leaky expression of the genes in the lac operon. So in biology, it's very rarely the case that anything is completely off or completely on, right? So you have a little bit, even in the off state, of this enzyme, beta-galactosidase, which is encoded for by the LAC-Z gene, present. So what is that? What is the meaning or the impact of that? Well, what that means is that you have a few enzymes of beta-gal inside the cell all the time, right? So if you're an E. coli cell and you're swimming around and you find yourself passing through some lactose, right? And some lactose gets into the cell. The question is, as that cell, you have a decision to make. Is it enough, right, to just see a little bit of lactose, right, and just de-repress this operon and start making the machinery? Or would it be more advantageous to make sure that you're under a condition of prolonged exposure to lactose before you really rev up the machinery, right, to begin to break down that lactose? And of course, the latter condition is the preferred sort of strategy. So the way that the cell achieves that is by using allolactose rather than lactose as the inducer. And what I'll tell you is that allolactose is essentially a side product of lactose breakdown, essentially in a isomerization. The details of the chemistry are not critical there, but beta-gal, in the presence of small amounts of beta-gal, the lactose will slowly be converted to this molecule allolactose. And so the reason why it's preferred to have or advantageous or has been evolutionarily selected for, right, to use allolactose as the inducer is that you will only build up enough of the allolactose molecule, right, if you're under a condition where you have enough exposure to lactose, what I would say, so side product of lactose breakdown builds up under prolonged exposure to lactose. So in other words, if you are a cell and you just are fleetingly exposed to this molecule, lactose, you're not just going to commit to a completely different metabolic strategy, right? You're going to wait until you fully have sensed your environment to commit, right, to this alternative strategy. And that's a little bit about why allolactose rather than lactose is the inducer. From a functional standpoint, maybe the most important point to take home is that binding of this inducer molecule to the repressor changes its conformation such that it reduces the affinity for the operator and it comes off, clearing the way for an RNA polymerase. But remember, under this particular condition, we haven't met our two necessary criteria for fully activating the lac operon. Because here you have lactose present, so you've de-repressed, right, the operon, right? So this reduces the affinity of repressor for the operator site. But if that's all that you do, that is not sufficient to fully turn on the lac operon. And so you will not get very strong transcription from this promoter until you're in this finally condition where you have low glucose, right? Again, that relates to the cyclic AMP second messenger levels rising, okay? They're accumulating. Coming back to that idea, what that does is it essentially allows this activator protein, that's the positive regulatory, right? So this is positive regulation, as was pointed out. Whereas the removal of the repressor by binding of the inducer allolactose is an alleviation of negative regulation. So let's just say removal of negative regulation. So it is definitely the case, I'm glad that it was pointed out, that we have both strategies enacted in a single operon. And now that cyclic AMP CRP complex can bind to the activator, that helps, you can see, through physical interaction with the RNA polymerase, much in the way that the sigma factor, as we talked earlier, helps RNA polymerase orient. Okay? This now improves the efficiency of binding and initiation, and now the system is on. Okay? So in this case, lac operon is fully on. Okay? So you have a lot going on here, right? You have to sort of understand the organization of an operon, what all those different components, the functions they serve, and then you have to understand how they respond under different, these 4 different combinations of categories of sugar availability that we've discussed, okay? So that's the lac operon. Are there any questions? Yes? Yes. So over here, the question was, is the repressor being constitutively expressed? And the answer, or constantly expressed, and the answer is yes, right? So there's no sort of feedback going on here. This is just being made, and then the question is, given the fact that repressor is always around, is it competent to bind the operator or not, depending on if it's bound to inducer or not, okay? And so notice here that you have both cases from that early strategy where you have some regulatory molecule, a small molecule binding, an effector molecule binding to it and removing it. And similarly, you have a small molecule in cyclic AMP binding to another regulatory factor and actually increasing the affinity for the DNA. Yes? CRP we can think of as like a housekeeping gene that is constitutively expressed. Yes. Okay. And I should also mention, along that line, that cyclic AMP is not a small molecule that is in any way specifically regulating only lactose, right? So this is a broad sort of signal of the metabolic state of the cell, and its binding partner, CRP, also, right, is present at many other promoters, depending on, sort of the strategy or the response needed to the metabolic state of the cell. Question in the back.
So in the first area, there was a low-level receptor.
Yes.
Does the polymerase still bind, but it just binds a very rare occasion?
Yeah, so it's basically a matter of probability of binding and promoter clearance, as we've discussed, right? So, and that becomes higher when it's given this extra affinity for the promoter through the activator protein CRP. Okay? That's right. Cyclic adenosine monophosphate. Okay? So you're going to find that often slightly structural, modifications or modified nucleotides serve as the second messenger molecules. And cyclic AMP is a classic case in point. Okay? Good. All good questions. Okay? This stuff is a little bit more fun, I think, to think about because it's much more conceptual, right? Then this goes to this, to this, to this, like the pathway of promoter initiation or something like that, which is also fun to think about, but maybe not as much fun. Okay. So that's the lac operon. Now we're going to talk about a completely different operon. And what's cool about this operon, so this is the operon that encodes the genes required for the biosynthetic pathway of tryptophan, the amino acid. And it also has multiple modes of regulation, okay? And so don't get, please don't get too bogged down in this, okay? But this is basically tryptophan biosynthetic pathway, biosynthesis, okay? It's enough to know that. And there are a bunch of genes required to catalyze these complex steps in tryptophan biosynthesis. And they are under the coordinated control of the tryptophan operon, okay? And so here we have a little bit more of a logical feedback type loop, which is this idea that when you have tryptophan concentrations in the cell that are high, right, we want to have the operon turned off. Right? So we don't want to be making more tryptophan if we already have enough, okay? And so in this case, it's really the opposite of what we just discussed with the lac repressor, right? Where actually the tryptophan itself, shown here, right, is the small effector molecule, and it's going to bind to the TRP repressor, which similarly is upstream, is expressed at a different site from the operon, in a way that's a manner that's similar to the lac operon. And so you have this tryptophan repressor protein, but its affinity for the operator, and in turn its ability to turn off the tryptophan operon is dependent on the binding of tryptophan itself. Okay? In other words, if you have plenty of tryptophan, right, then tryptophan will bind to your TRP repressor, and that will engage the operator. And this is another example of negative regulation. Okay? So that's one level of control, which I think is pretty straightforward to understand, right? It's a pretty intuitive feedback loop. If you have tryptophan already, you don't need to make more, and so that directly feeds back into the repression mechanism, okay? But as is often the case, that's not the whole story of tryptophan regulation. And in fact, there's a really interesting additional layer of complexity to the tryptophan operon, which relates, I can start to describe it here at a very sort of broad level, which relates to the fact that at the front end of the coding region, okay, so here's our promoter, here's our operon, which is currently being shown in the repressed state with the tryptophan repressor bound to tryptophan and occluding the promoter, or blocking the promoter from being used by an RNA polymerase. Then you have all these genes named TRPE, TRPD, so on, TRPE. Yeah, okay. So, but upstream of TRPE, right, is this so-called leader region. Okay, so remember, promoters here, the RNA is going to be transcribed starting right up here, shown by the green. So there's a region upstream of the actual individual coding regions of this polycistronic multi-gene RNA that doesn't necessarily encode at all for any of the metabolic enzymes needed for biosynthesis of tryptophan, but it's actually a component of the regulator itself, which includes a specialized sequence called the attenuator. Okay? And I think that this is a really interesting mechanism of regulation because it relies on our knowledge of several different things that we've discussed so far in the class, right? So if we zoom in on the RNA that is made, okay, upon activation of the transcriptional process that is happening at the operon, the tryptophan operon, then we see something like this. So this is the beginning of the RNA, and just to orient you, I'll just flip back for a quick moment. So we're basically looking at what is happening to the mRNA in sort of this region right here. So we're going to zoom in on what's going on both from a sequence and a structure perspective in that early region, the so-called leader peptide region of the messenger RNA. So the way that this works really relies upon the underlying foundational understanding, right, that in bacteria, which we've talked about, transcription and translation are coupled, right? So these two processes are happening at the same time in the same place. I've really said that a number of times, right? Okay. So now here is a regulatory mechanism that absolutely relies upon that being the case. And so why is that? Okay. So the basic concept here is that if we look at the organization of the leader region of the messenger RNA, we can identify some critical segments. And the way that that's done here is basically there's a coding region. So notice here there's a start codon, AUG, right here. Okay? And that is not actually going to encode for a full polypeptide or a gene in the formal sense, but rather it's just encoding a very short peptide. Okay? And a critical component of this so-called leader peptide, which is in a sense an important regulatory element of the tryptophan operon, is that this leader peptide has in it several codons for tryptophan. Okay? So hopefully maybe something is starting to click in your brain right now that, well, maybe this is some way of sensing, right, by the ribosome now, if there's enough tryptophan around in the cell, right, by making a peptide that has tryptophan in it. Okay? So that's the first segment of the leader peptide that you have to be familiar with to understand how this works. We'll come back to that. And then you have these other regions of the RNA which seemingly in an arbitrary way are being designated number 2, 3, and 4, with the leader peptide being region number 1. Drawn this way, it is somewhat arbitrary, but the reason why it's drawn this way is because it's in fact not arbitrary, but rather relates to potential structural properties that this messenger RNA can assume. Okay, different structures that this RNA can fold into. And the main concept or take-home here is that you have this region 3, okay, and region 3 can, let me just write this as clear as I can. So region 3 of this leader can base pair in a mutually exclusive fashion, okay, with either region 2, which is upstream, or 5' to it, or region 4, okay? So it can base pair with either, I really want to stress that it's mutually exclusive, region 2 or region 4. Okay, and I'll show you a picture of that in a moment. Okay, so at this level, all we need to know about how this is working is that there is obviously not enough tryptophan in the cell for this mechanism to be repressing, okay, because we're making the RNA. So we have low enough tryptophan that we have essentially de-repressed at the operator. We've started to transcribe, but now we have like a fine-tuned mechanism to ask the question of, do we really want to kick into full gear transcription of the entire operon, right, that has all these different genes for tryptophan biosynthesis. And so how does this work? So here's the picture that actually allows you to understand how it works. And I think, and I should point out that I'm discussing this for tryptophan, but this is true for a large number of amino acid biosynthetic pathways. So this general paradigm or concept applies to many different amino acid biosyntheses. Okay? And so what's going on here is the following. So here we have the same color coding as I just described of the messenger RNA, which is being transcribed from that leader region. And so here's this idea, so starting from this important concept of co-transcriptional translation. Okay, both are happening in a coordinated fashion. So you have transcription occurring over here and translation over here. And here's the neat thing, which is basically as the RNA is being made, that initial start codon for the leader peptide can be recognized by the ribosome and translated. Okay? So it's a short peptide. And if it can be translated efficiently, then what ends up happening is that the ribosome can move through this leader peptide coding region, which includes, importantly, several tryptophan codons. Okay? So if there's enough tryptophan in the cell to allow efficient synthesis of this so-called leader peptide, then the result of that is that the ribosome actually influences the structural properties of this RNA, in the sense that it basically, what I told you before is that region 3, shown here, can pair in principle with region 2, which I'll come to in a moment, or region 4, shown here. But in the case where tryptophan is at sufficiently high levels, the ribosome blocks a segment of region 2 and prevents the pairing of 3 to 2. Does that make sense? So as transcription is moving in this direction, the efficient translation by the ribosome allows it to alter the folding property such that this 3 region instead binds to the region 4, and hopefully this picture looks somewhat familiar to you. Okay? Does it look familiar to you? Okay, good. Why does that look familiar? So that looks like a terminator, right? Not in the Arnold Schwarzenegger sense of the term, okay? But in the transcriptional terminator sense, right? So this is a hairpin followed by a poly U, right? So this is row-independent termination, right, at work in a completely different context, to what we've discussed up to this point. So this is the so-called attenuator structure. So tryptophan in the cell is high enough to allow efficient synthesis of the leader peptide such that regions number 3, this is of the mRNA, and number 4 can form a hairpin. This attenuator or terminator hairpin can form an RNA hairpin and terminate transcription early. So this is like a backup mechanism to turning off tryptophan operon expression if there's enough tryptophan present. So, and it's an RNA structure mediated one, okay? Now, in the other circumstance, okay, so is that clear to everybody? More or less, the concept is reasonably clear. So in the other circumstance where tryptophan is low, really low, okay? So if tryptophan is very low, okay? Now what's going to happen when we begin to translate this leader peptide? Well, it's going to get to the codon for a tryptophan incorporation, right, during translation. And it's not going to have any charged tryptophan tRNA around to incorporate the appropriate cognate amino acid. So the ribosome is going to stall, in this case, waiting, saying, where's my tryptophan? Okay, right? So the ribosome stalls here. So in a sense, the ribosome is the sensor, right, of this attenuation mechanism. It's determining whether or not there's enough tryptophan to make more or not make more, which is kind of cool to think about, that the protein translational machinery itself, right, through modulation of RNA structure, can actually regulate the expression of a particular cluster of genes. So that's like heavy stuff, right, for me at least. That brings a lot of concepts together into a functional paradigm. And as a result of stalling here in the leader peptide region, that allows, during transcription, right, the 2,3 hairpin to form, okay, as opposed to the 3,4. And as a result, this 2,3 hairpin prevents the formation of this attenuator and releases the RNA polymerase to transcribe the rest of the operon. Okay? So you have this switch, right, on and off at the RNA structural level, which is being controlled by the ribosome. Yes?
So is the 2,3 hairpin just like more stable or more likely to form?
So all things being equal, well, so there's a couple factors that go on there. One has to do with the fact that the question essentially becomes, and so I don't know the answer about if you just took the 3 regions and put them in a test tube, you would get some dynamic equilibrium between the two. But the way this control is controlled is kinetically. So the question is basically, has the ribosome caught up to region 2 by the time that region 3 has been made by the RNA polymerase? If it has not, and region 3 is shunted out of the exit channel of the RNA polymerase, it is free to form this 2,3 hairpin first, before region 4 has ever been formed. Okay? And by the time you maybe are thinking about, well, then what happens now, right? By the time you might have some dynamic equilibrium between maybe those two states, that RNA polymerase is gone, right? It can't be. It's past that terminator sequence. It's already moved on. So it's a kinetic control mechanism that favors this particular hairpin. And I don't know, it's a good question if all things being equal, if the actual energetic stability of this hairpin is greater. I don't know the answer to that. But the way the book describes it and the way you should think of it is that it's kinetically controlled through co-transcriptional translation. Okay? Okay? So that's another concept of gene regulation. It's got quiet. Yeah.
So the ribosome still stalls when there's a 2,3 hairpin.
The ribosome still stalls. Here, you mean?
Yeah.
So what is actually so the ribosome stalling is actually what is allowing that 2,3 hairpin to form in the first place, right? Because if the ribosome didn't stall, right, then it gets into the region 2, the base of this hairpin, and when region 3 comes out, it can actually nucleate that structure. Okay? Does that make sense? Okay, great. All right, so that's the tryptophan operon. Okay, and that, again, ties in a lot of concepts. So now let's talk about another example of gene regulation. Oh, not yet. Good question, right? So a different ribosome would do that, okay? And that's because each of, it's not shown in this figure, or this figure, but each of these genes, right, has its own Chignal-Garneau, its own start site, and so on, right? And that's an important question. So it's not like it's the same ribosome that's scanning through this giant RNA, okay? Good. Okay. So now here's another interesting subject of gene regulation. This one relates to the idea of feedback of the biosynthesis or translation of the actual translational machinery itself, the proteins required to make the ribosome. Okay? And so what's interesting about many of the ribosomal components, right, here, is that they are regulated in a really distinct way. So what we have here are a series of different, you don't have to worry about memorizing all the details here, but a bunch of the different proteins that go into building the ribosome. So just to orient you, so the letters L here refer to large subunit. So L10 is a protein of the large subunit. S12 is a protein of the small subunit, and so on and so forth. And you can see, like, for example, the S10 operon is a single operon that encodes, what, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11 or so individual ribosomal genes, okay, proteins that are going to code for. The feedback loop here is an interesting negative feedback loop. Where you have a paradigm, and it's not obvious here. So what's meant to be shown here, so what's being drawn here is not the DNA, but all of these are RNA. So this is a unique, so far what we've talked about, right, is we've talked about activators or repressors binding to operons at the DNA level and impacting transcription, right? In this particular feedback loop, we're talking about the efficiency of translation once an RNA is already made, okay? So now we're having RNA binding proteins binding to some RNA, right, and influencing their ability to be translated by the ribosome, okay? So this is being regulated at the translational level. So you can see that there's a lot of diversity here. And what's interesting, or sort of clever about this feedback, is that in each, so if we just pinpoint one, let's just focus on one of these operons. So obviously the ribosome is a major mega machine, right? It has lots of different parts that are required. And in fact, the amount of ribosome that is being made and assembled relates very much to the metabolic state of the cell, as you might expect. So as cells are growing very rapidly under rich growth conditions, in order to achieve that growth, they need a lot of ribosomes. So at maximal growth potential, the ribosome can represent up to 40% of the dry weight of an entire cell. So there's just a lot of ribosomes when a cell is growing really vigorously. But you only want to make all those ribosomes when you need them, right? And so there is a need for regulating all of these different components, both the RNA and the DNA, the protein components of the ribosome. So if we focus in on S8, so just to give you a sense, so this is the small subunit 8 protein, okay? Just so that you get a sense of what that S8 means, okay? And essentially what S8 can do, if there's, in any of these cases, if there's free S8 floating around the cell, then what that suggests is that you don't have a lot of ribosomal RNA waiting for the ribosomal proteins to co-assemble to make a ribosome. Okay? So the whole sort of take-home of this feedback loop is anytime you have feedback, or I'm sorry, excess of these proteins that suggests that you don't have enough RNA, and you need, I should say, ribosomal RNA. Okay? So the presence in any of these cases of excess ribosomal proteins, so that's all of these, right? That indicates that, what should I say, ribosomal RNA, try that the way you were, our RNA production is, let's say, limiting, okay? That you don't have enough ribosomal RNA to sop up all these ribosomal proteins in the cell in the form of making ribosomes, okay? So when the cell is under a condition where there's not enough ribosomal RNA, it doesn't want to keep cranking out ribosomal proteins, right? And so what happens is, as soon as that's the case, then these proteins can come back and negatively regulate by binding upstream and preventing ribosome translation of these RNAs. So these proteins can bind to mRNA and negatively regulate translation. Now, admittedly, it's a little confusing, actually, relating to the question that was just asked about how each of the ribosomes is loaded on and each of the genes. So the paradigm, which is true, is that each of these essentially has its own entry site for a ribosome. So how is it that binding of S8 up here is going to shut down translation of this entire RNA? And the answer to that question is not well understood. And it's thought that it has to do with the global structure of the RNA and things that are sort of not only beyond the scope of this class, but we just don't fully understand at this point. Okay, so just not to be confused here. So the take-home here is that these ribosomal proteins would certainly prefer to bind to their appropriate site on the ribosome, right? So the affinity, this is the last point of this topic, the affinity of the ribosomal proteins for their appropriate site on ribosomal RNA, rRNA, is greater than for any of these mRNAs. So if given the choice, right, these freely diffusing ribosomal proteins are more likely to end up in a ribosome if there's a ribosomal RNA that needs it, right, than on these mRNAs to negative regulate the production of more ribosomal proteins. Okay? So that's the competition there, is whether this free protein should bind to an mRNA or to a ribosomal RNA. And will always bind to the ribosomal RNA if it has the chance. But it's only when there's not a ribosomal RNA present that it then will come back, and because that's then in itself, that is a sign or a signal that there's not enough ribosome RNA to make more ribosomes. We should shut down ribosome protein synthesis, and it goes back and negatively regulates that at the translational level. Okay? Does that make sense? There's a lot of different concepts. I recognize that. I recognize it so much that I think the next concept I'm going to skip, because I can see that you guys are getting a little saturated right now. And so there's another thing called the stringent response in bacteria. It's quite interesting, but I'm not going to describe it to you because I feel like I've given you a ton of information already. But we're not done yet, so don't pack up. Okay? Because there's one last mode, so I'm skipping over the stringent response. So take note of that. But the last thing I would like to point out is this, is that it's not just all about proteins regulating gene expression, but we now know that small RNAs can actually also serve a similar function. So it's not just about proteins binding to DNA, or in the case of the ribosomal feedback, proteins binding to RNA, regulating gene expression. But we can also have mRNA expression through the action of small RNAs. And so, again, details are not critical here, but concepts are what I'd like to sort of stress here. And what's going on here is that you have a small RNA, okay? And what it can do is that the sort of basal state of this particular messenger RNA, so this is an mRNA, okay? So in this top example here, notice that there's structure, and really the main concept here, again, is this idea that RNA structure can play a very significant and central role in gene expression regulation. And the exact way that happens at different genes is, very actively studied. So RNA structure can play a very important role in gene regulation. So that's sort of a broad point. And what's happening here is notice that this ribosome binding site, that Shine-Dalgarno site, is tied up in the form of RNA structure, and so the ribosome can't get on there, right? And then what ends up happening, this is somewhat similar to the tryptophan operon, except now instead of an RNA that's acting internally, you have an RNA that we use the term acting in trans. So when we say that a factor acts in trans, that means that it is diffusing in, right? RNA is, in this case, acting in trans, diffusing in to interact with a target shown here. Okay, it does have help from a protein, but let's just focus on the RNA. And what the implication of that interaction is it disrupts this hairpin and frees up the ribosome entry site so that translation can occur. So this idea of small RNAs acting in trans is essentially to be contrasted with RNAs acting, what we would say, in cis, which would be similar to what we discussed for the trip operon, where structures are all within one RNA, right? That's the term in cis, okay? Different than what we're used to thinking in chemistry of trans and cis, right? So this is a different concept, right? So, but the key point is that you can activate again at the translational level. So small RNA acting in trans can alter RNA structure to facilitate translation. Okay. And there are other, obviously, examples of how this can work, but I don't want to bore you with too much detail. Okay, so the key point that I wanted to make sure to include is that up to this point, everything that we had discussed, right, had to do with the idea of a protein coming in and acting as an activator or repressor. But it's very important that I leave you with the message that RNAs, small RNAs in particular, can also serve this function, right? And there's like a budding and an explosion of research right now investigating how is it. It turns out that much of our genome, which was formerly thought to be just quiet or untranscribed, is in fact transcribed into what are now being collectively referred to as these non-coding RNAs. There is this vast sea of up to 100,000 different non-coding RNAs in each of our cells, right? And we don't know, we might know what like a half a dozen of them do, 6, right, out of 100,000. So it's really uncharted territory to begin to understand, well, maybe some of these RNAs are actually playing specific roles as transcriptional or protein expression gene regulatory mechanisms, okay? Whew, it's a lot of stuff to talk about. With that, I'm done now. I really want to thank you guys for being a great class this quarter. I don't like to get too sappy, but I will say that this is a great class to teach 100A for a number of reasons. You guys are great students. The material is just getting you started now. So I feel like a proud papa in a way because I've now hopefully set the stage for what is a very rewarding year of biochemistry ahead. So I wish you luck with that. Thank you. I appreciate that. As long as you're in the clapping mood, let's give our TAs a hand as well. as well as our MSI instructor. Okay. Thank you guys so much. Good luck studying with finals. See you later.